using software testing techniques to infer biological models. Systems biology is an interdisciplinary field in which a system is abstracted into a whole rather than individual parts. For example, pictured here is an anaerobic digestive tank. As a whole, the system can convert various inputs, such as bacteria and landfill waste, and produce methane gas that could be used to power local homes and businesses. Now, if we look inside this integrated system, we see some important parts, such as the organisms called methanogens that perform this degradation of the inputs. And inside these methanogens are the underlying metabolic networks that provide this functionality. Let's take a closer look at these metabolic networks. In my work, I have focused on microbial organisms whose metabolism can be modeled using a network. In these networks, or graphs, the nodes are compounds and the edges are chemical reactions. The network shown here is for E. coli from the KEGG database. Say, for example, we choose one input environment. This input will cause certain reactions in the network to occur, causing a specific output behavior, such as a high yield of methane. But under a different input environment, a different set of reactions is activated, causing perhaps a medium yield of methane. And under a third environment, we might see a low yield of methane. Let's consider how many total input environments we could have. If we had 7 binary factors that could be in the environment or not, we would have 128 possible input environments. But if we scale this up to 20 factors, this jumps to over 1 million input environments. The reason for this sharp jump is that the combinatorial space grows exponentially. Therein lies the problem. There are simply too many input environments for us to study. There are many potential benefits stemming from systems biology, such as optimizing the production yield of a plant like the digestive tank, revealing new chemical interactions leading to medical discoveries, engineering a new biological system of our own design, and improving computational models. For this project, I worked in an interdisciplinary team that approached the problem from a software engineering perspective. There is a lot of work in software testing of programs with many input options called configurations, just like in systems biology. I applied methods that originated in the software testing research, such as sampling and classification, which have been used together in related work to reduce the cost of testing. Putting these ideas together, we present the process BioSimp, which stands for biological sampling, inference, modeling, and prediction. The process starts with choosing which environmental factors we want to vary. In a case study, we looked at 2 different gut microbes found in the human body. Changes in the abundances of these organisms has been linked to nutritional-related disorders such as obesity. We chose 7 different chemical compounds to vary in the environment. Each of these can be turned either on or off, leading to 128 different input environments. The next step is to sample this combinatorial space in case it is too large. As we saw before, the combinatorial space of possible input environments explodes exponentially. However, by using methods of sampling, such as combinatorial interaction testing from software engineering, we can significantly reduce this space to a logarithmic function. Next, we run experiments on these chosen samples. In this work, our dependent variable, or phenotype, that we study is the growth of the organism. It either grows or it doesn't grow. Our collaborators ran experiments in the wet lab to understand what occurs in nature. I ran experiments in simulation to explore the potential of the computational models. I became an expert user of the Department of Energy's Systems Biology Knowledge Base, or KBase. By using their SDK, or Software Development Kit, I was able to script up parallel runs on a supercomputer. Next, we take the output growth data and try to classify the results using inference to learn what are the influential environmental factors. In other words, which input factors affect the output and how. Here we see the classification tree for B theta on the laboratory data. We can see that glucose is the most important factor. Without it, B theta will not grow. The next most important factor is hematin. With it, B theta will grow. And without it, it depends on the interaction between vitamins K and B12. So note here that we started with 7 factors and reduced it to only 4 that influence the growth of B theta. We can use all this knowledge to understand how the environmental factors affect the chemical reactions in the metabolic models. Using the data from KBase, I was able to build a reaction coverage map showing for each reaction if they are being executed, and if so, in which direction. Another interesting result is that I was able to condense all 128 input environments into only 14 patterns of reaction execution. Finally, we can put all this together to make predictions on new, unstudied environmental conditions. One insight from this work was the discovery of this unknown interaction in B theta between vitamins K and B12, which has a potential to impact research on obesity. Our collaborators are currently running laboratory experiments to investigate this potential relationship. I also discovered that 75% of the reactions in the metabolic model don't change under the environmental conditions we tested. Then we see 25% of the reactions vary. These are the reactions that identify significant pathways that could be manipulated in order to control the output. I also brought Biosim to the broader community by implementing the workflow into KBase as a narrative. KBase's online interface is open to the public and has thousands of users. Now this workflow can be used by members of the community. Users can simply copy my narrative and import their own data to make their own discoveries. I would also like to thank our collaborators, as this was a very interdisciplinary project.